Hi everyone, welcome to the second episode of my tutorials on circles for bioinformatics. Right. So for this tutorial, this is what we are going to. We are going to use circles to generate this cool plot here. I'll show you how to do everything. All right. So all the example data and input files, I will show you where to get them there for free. Okay. Now, before I proceed, let me say that the example codes I'm going to use, they are all here. All right, so these are the codes I'm going to use. So if you want the codes, all right, if you want the codes, you can check the description of the download link to get them. It's there. All right, so just check the descriptions for it. But I'm going to go through the code line by line with you. I'm going to do this in an interactive session and explain what each of these lines does. All right, so I'm doing this because beginners will need to understand how circles work with respect to the commands that you use. So that's what we are going to do. Now let's continue. So before we start the exercise, I'll explain a couple of things here with respect to the plots we are going to generate. So the plot we are going to generate shows SNP distribution. All right. So I got this data from by calling variants from identify variant in a Plasmodium falciparum sample. All right. So I just took the FASTQ files and then performed mapping and then generated the variance data. That's VCF file. And then from the VCF file, I extracted the information to generate this plot here. Of course, if you want more tutorials on how to generate these kinds of plots, you can always reach out to me. My contact is in the description box. So let's look at the image or this plot here. So we have the ideogram, which is here. I will show that right now. Okay, so let's just continue. I'll first enable the arrow and then I will continue. All right, so let's proceed. So I'll go over again. So the ideogram is this one here. So this the outer one is the ideogram. I will explain it very soon. And we also have the labels here showing the chromosomes. All right, so we have chromosome 1 to 14. So here we have 4, 5, and then 6. And we also have the SNP distribution, which is the plot itself, which is here, the data that we want to represent. So it's the SNP distribution. And here I'm using the length strategy. I will also explain that later on. All right, so this is the ideogram, trying to break everything down for you to understand because some viewers may be beginners. So there's the ideogram and it's used to depict the chromosomes. Okay, it can also be used to depict or show or display the sequence context, for example, but it depends on what data you are presenting or what data you want to visualize. So for in the context of this tutorial here, the ideogram here represents the chromosomes. Okay, so from chromosome 1 to 14. So this is an unlabeled one, but you can also label it like this. So when you label it, it becomes very easy to interpret your data. And of course, you can see that each of the chromosomes has a different color. So for the color types, you will decide what color to use to represent your chromosomes. And you can also see that the chromosomes have different lengths. Okay, so those lengths are reflected in this image here, but you can also do a reordering. All right, there are things you can do, but for the most part, just displaying them like this will also work fine for you. So those little details, that one I will explain them when there's the need. But of course, you can also check the Circos documentation page and you'll find more information. So just make sure to check the description. You'll find links to tutorials that will help you get started with respect to Circos. All right. So for ideograms, they are generated from character files. So character files are something like this. So it defines the chromosomes that you want to display. So this is a chromosome, and then we have the chromosome name and then the label. Then we also have the start and end, and there's also the color. All right, so this is how a typical character file looks like. So this is the name label. So this label is what is shown. Let's just go back. This is what has been shown here, 1, 2, 3. All right, so you see them here, 1, 2, 3 up to 14. All right, and we have the start and end, and then the color. So that's how a typical character looks like. But of course, with the character, I've also prepared it. So I will show you where to download that as well. All right. So you just go to the SNP distribution. That is this one here. So there's the SNP distribution that we want to represent. And then, so finally, this is what we will get at the end of the day. We combine both the ideogram and the SNP distribution to get something like this. So this is what we are going to generate at the end of the day. So what you need to do is to make sure you have Circos installed. So make sure you have Circos installed, and then you can follow this tutorial. And of course, if you have any challenge with installing Circos, you can always let me know, and then I will try to help you. So you can book a session, and then we can look at that together. So without further ado, let's get started. So what we are going to do is to first download the example data. So let's go to the GitHub repo, which has the example data. So this is the GitHub repo, which has the example data. So I'll leave the link to this page in the description box. So here you need to look for this one here, Circos episode 2 input files. This is a file here. That's what you need to download. All right. So when you're on this page, click on it and you'll be sent to another page like this. And when you are here, you can click on the download button to download. You can also use RAW if you want to download from a terminal. So all I have to do is to right click and then go to copy link, and you can download via wget. All right, I've shown how to do that a countless times, so just try to download them. So click here to download, or use the download link here and then download on the terminal. All right, so that's what you have to do. So once you are done, you can extract the contents. I have already downloaded the file myself, so we will go take a look at it now. All right, so this is the input files that we are going to use. Okay, so I'll extract mine here. You can also do it on a command line, by the way. If you have Linux, that's great because it helps you to do things easily. But if you're also using Windows, you can just extract the content. So that's how we do it. Let's just extract the content. So this is the output that will generate, and these are the files. So here I have the karyotype and I have the link file. I'll start with the karyotype here. I think I had already opened it. I'll try to open it here. Yeah. So this is it. So we will use this to show the ideogram, and then we are going to also generate the SNP distribution using the link file here. So the link file contains information that we are going to use. All right, so that's what we are going to do. So make sure you have Circos installed, and then you can proceed. Okay, so now it's time to start. So we are going to write a Circos script, or let's say Circos script, to help us do this. Okay, so let's proceed by writing our script. Now what you need to know is that the script is just an ordinary text file. So in this context, you can just open a new text file and then just write the quotes in it, or you can also use the terminal, all right, to do the same thing. I'll open a terminal and then start. But you can use any text file to also create the script. It's just a text file. Let me just go over again. So it's just like this, a typical text file like this, and you can just start typing. That's what you need to do. All right, but sometimes you'll be working on a terminal on the command line. Okay, sometimes you will have a system where there's no text file, so you need to write everything on a terminal. So that's why I'm going to use the terminal to do that. All right, so just take note of that. Okay, so we can proceed. So I'll do this. I'll open a terminal here and then I'll start. All right, and then we can proceed. Yeah, and before I even do that, let me say that. So the link file here, all right, this information I obtained from this VCF file here. So this is the VCF file I was talking about, the SNP data. All right, so I extracted the content, use some other scripts to process it, and then get this data here. All right, so for the data, you will decide how you want it to be represented. So that means that the scripts you write will determine how this information will be displayed or even arranged or ordered. All right, so if you also have a similar data and you want to generate the Circos plots, you can also reach out to me. We can have a session and I will show you how to do all of these stuffs. All right, so just take note of that. So let's continue. So we will start. Now with Circos, we will need to first generate the ideogram. All right, and so that means that you will have to write a code to display it. Now I'll take you to the Circos page itself. So this is the Circos page, one of the pages, one of the tutorials. Okay, I'll leave the link to this page in the description box. So this shows how to generate the ideogram. You have to first get your type. Let's just look at something here. We will do everything, so don't worry. I just want to show you how a typical one looks like. So this a typical one, we displayed something similar, and then there's the configuration. All right, so the configuration shows how the ideogram will be displayed. All right, so we are going to do something similar here. All right, we will do something similar here and then proceed. So this is the ideogram conf. We write all our codes like this. Don't worry, I will explain everything. We write all our codes like this and then we will now use it. All right, so that's what we are going to do. So I'll move back to the terminal. I think I'll do it this way. Let me go back and I'll create my ideogram.conf file. Let's just go back here. So we are going to create something like this. So ideogram.conf, and then we'll put in our code here. All right, that's what we are going to do. So ours will be slightly different from what is here because I want to make it very simple for beginners to understand. But if you still want the already written codes, just check the description box. You'll find the link to get the scripts. All right, so let's proceed. So we are going to write our ideogram configuration file. We will create it. So I'll use nano and I'll say ideogram.conf to open it like this. All right, and then I'm going to start. So to start, you need to bring this less than symbol here, and I'll say ideogram. This is the rule, by the way, so I will not be able to explain some things here because these are rules. It's just like you having your language, there are words you have to use, there are rules. So these rules, you may not necessarily know why it has been given, only the creators know. So for some of these symbols, that is the rule that has been given, so I can only write it as it is. All right, so that is, so that's how we define the ideogram. So we write it like this, and then we come here, we also put this one there to end it. So we say ideogram. I'm going to type it here, so it's like this slash, I'll say ideogram. So this is how we start and we end it. All right, so we need to take note of that. So now we can proceed. We can start with the actual ideogram codes, but let's just confirm here first. I usually want to check it. Yeah, so you can see this is the start, and then this is the end. All right, so that is what we have done here. And then in between, we will put in our quotes. That's what we are going to do next. All right, so let's proceed. So let's start. So we will say show equals yes. That means we are going to display the ideogram. All right, and then we are also going to demonstrate, or not demonstrate, we are also going to set the spacing. All right, so in the next line here, we are going to say this and say spacing. All right, and then you say default equals 2u. Okay, so here we are setting the spacing between ideograms. All right, that's what we are doing here. So I'll just say spacing between ideograms. Okay, so here when you see a line beginning with hash, it's a comment, it's not part of the code, so it's used to give extra information about that line or group of lines. All right, so here I'm setting the default spacing between ideograms to 2u. Then I'll also end this one. I'll say spacing. That means I have ended that particular configuration or settings. All right, so this is all it's right for the spacing option or configuration. Now we also need to set something else. So we have ideogram position thickness and fill. All right. Now, with some of these values, it's not specific. All right. It will depend on the image. So sometimes you have to do some experiments. You have to start with some values, and then when you view your plots, you can make some modifications. All right. So these values may change depend on the amount of information you want to represent. But all the same, it's good to have a starting point, and that is what we are doing here. So we will do this. I will say radius equals 0.90r. That's for the ideogram. And then for thickness, I'll say equals 20p. Then for fill equals yes. We are going to fill it. And then show label equals yes. Remember, we show the labels chromosome 1 to 14. So 1 to 14, that's the label. So we are saying yes because we want to show that. And then I'll say label font equals default. So this is it. All right. Now these values here, I picked it from one of the Circos tutorials, and then I used it on my data, and it's worked. So that is why I'm also using these particular values here. All right. So many a times you will have to get the values and then use that to plot, and then it may happen that the values work fine for you, but if it doesn't work well, then you can either increase or decrease those values, or maybe specify or omit some of those options here. So these things, they are still experiments, so you have to try, and then hopefully you get it. And with Circos, you just have to keep on trying and experimenting, and everything will become very, very clear to you. All right. So Circos is not that difficult if you understand the structure and the system. All right. So just take your time and then follow. Of course, I'll be making more tutorials on Circos, so you need to make sure you come back to this channel and then watch. And of course, those who want other tutorials or one-on-one sessions too, you can let me know, and then I will try to help. All right. Okay. So that is it for the ideogram position, thickness, and then the fill. Now there's something else we need to do. Let me just move down and then check here. So we need to set label radius. All right, so the label radius to help us to specify the space between the labels and then the outer ideogram. I'll just put that also here. So specify a space between the labels and the outer ideogram. Here referring to the chromosomes, those that are outside, okay, the outer ones. So we will set it to 50 pixels outside the outer ideogram. All right, so the ideogram itself has its radius, and the labels also has their radius. All right, so this is how it's going to be. We will say label radius equals dims ideogram, radius outer, we say plus 50p. All right, so in this case, we said 50 pixels outside the outer ideogram radius. So outer ideogram radius, and then we add 50 pixels to it. That's 50p. That's what we've done here. And then the label size equals 40. All right. So that's what we are going to use. Of course, if it's too small, you can always change them. All right. So this is it. All right. So now that we have this, we will now proceed, all right, to do something else. So let's continue. We also set label equals yes to kind of format it properly for us. Okay, so this becomes the final command. All right, so now we can proceed. Let's just clear the rest of the lines to get this. So this is the final command here. All right, so now we have our ideogram.conf. So this is it. So now that we have this, we can now close it. I'll just go over We just need to make sure that we have everything we need here. I'll go over everything and then the comments as well. So I have them here. So now I can exit it, and then that's it. All right. So of course, if you look at it here too, now it has been created. This is it. So that's all that we need. All right. It's very simple. So if you don't want to use the terminal, you can also use a text file here like this, and then now go to save as and then save it. That's also possible. So you can create your new text file and then also do your commands like this. All right, and then put everything you want there, then just end it. All right, so that's also possible to do. And then you can now go to save and then save as, and then that's it. But I'm choosing to do it on the command line. Okay, so this is my ideogram.conf, which is now here. Okay, so this is it. So now we have the ideogram. So after doing that, we also need to add a configuration file. So the configuration file is actually the master script. So the configuration file is supposed to be used to aggregate all the other scripts and also specify some key information that will help us to generate the plots. So we are also going to create that configuration script. All right, or configuration file. So I will do that here. So I'm going to say nano, and I'll say circos conf 1.conf. Okay, because I'm going to create another configuration file. So I'm just using circos conf 1 so that it differentiates what I'm going to create later on. So there's the first configuration file. And let me say that the name here, you can also change it. You can use a different name. That is up to you. But I choose to do this because I always want to keep track of everything that I do. So I say circos underscore conf underscore 1.conf. So this is the name that I'm using. But I repeat, you can always use a different name. So you can use any name you are comfortable with to save it. All right. So there is it. So now we are going to do this. With the conf file, we are first going to do this. You see, you look at the chromosome name, size, and color definition. All right. So that's it. So that one we have already created it. That's the karyotype file here. So what we are going to do is to import it here. All right. So I'll say karyotype equals, and I'll specify it here. I'll say karyotype.txt. Okay. So I'll say karyotype. Let me just do it again. So it's karyotype equals, I'll say karyotype.txt. So this is how I am defining it. All right. So let's go back to the karyotype. This is it. I am giving it the exact same name because that's how it's supposed to be. And then because I'm going to be working in the same directory as where this one can be found, the character.txt, that is why I chose to just give its name. But if the file is in a different directory, then you have to specify the full path. All right. So usually I would advise that you put all your configuration files in the same directory that you are going to execute Circos, so that you don't have the so you don't go through the trouble of specifying the full path for these individual files. All right. Okay. Now I'll define the chromosome unit. So chromosome underscore units equals, and that's what I'm going to use. I'll say 1, 1, 2, 3, 1, 2, 3. That's 1 MB. All right, that's what I am going to use here. So these are the units. So the units are 1 MB. Okay, so let me just go through again to make sure everything is okay, and then we can proceed. All right, so that has been done nicely. Okay, so now we can proceed. Let's do this. Now we also need to import the ideogram, and then we will proceed. So I'll also import the ideogram. And of course, instead of importing the ideogram configuration file, you can also manually type everything here like this. But then sometimes when you have all the codes in one file, it can be cumbersome, all right, especially if you're going to edit it. So you put them in separate files and you can import them. All right, so that's what I'm going to do. So I'll just say, if you want to import or include a file, you put it like this and you say include. Now say ideogram.conf. I have included that one here. Okay, so that is it. Now we need to also include some additional files which are always mandatory, and one of them is the housekeeping.conf. Now for the functions of these files, I'll leave the description. I will leave a link in the description that you can use to read more about these files. Okay, so all the information for these other mandatory files can be found on the Circos tutorial page. So I will leave that to you to check. So we'll include, let me just also include housekeeping.conf. We need to include that. And then we also need to include the image conf. Okay, let me just put it here. So housekeeping configuration file, or let's just configuration file. And then you also need to include the image configuration file. So that is what include, let's say image.conf. Okay, so that's it. But with this particular one, the rule changes. All right, so before we even do that, we need to specify it's like this. We need to say dash image, because it's a block, and then we now include our image.conf, and then we end it like this, image. So this one is different from this, slightly different from this. So we put everything here like this. All right, but you can always experiment on certain things to see how they go. All right, so that's how I'm going to put it. Okay, so these are the mandatory files that you need to add. It's always important. And then you also need to include another file. So I'll put that also here. So we also need to include the colors and fonts pattern configuration file. Okay, so that's one is here. So that one too, we see include colors fonts patterns.conf. We need to also include that file. It's very important. Okay, so we have a total of 3 configuration files here. Okay, so that is done. So let's just go over again. We first import our character type file. Let me just add that also here. Or in Circos, we even say include. Okay, so we can use the word include or import. That's fine. But I chose to use the word import here because we are importing them, by the way. So I have that everything here. So this is how the configuration file will now look like, the final one. So we can now exit and save the changes. Then we can do an ls to make sure it's there. Of course, if you have a GUI, you can still check and make sure it's there. So this is it. And so this is what I'm going to now execute. All right. So I'll do this. I will say circos. I'll say -conf. And I'll say circos conf1.conf. So I'm going to execute this command. Right. So that's how it's going to be done. Right. So that has been done. Okay. So once that's been done, you can just check it. If you're on Linux, you can do an ls to check the files which are here, output files. But if you're using your GUI, then just go to that folder and we are going to find the file. So we have the PNG format and the SVG format. So by default, Circos generates a PNG format for you and then an SVG. But you can always disable the SVG option. That's something you can also do. But for this tutorial, I'll just generate both. All right. So SVG, you can always edit and add or subtract information. So I normally generate both files and then I decide what to do with them. So this is it. So this is the final output file that we have here. Okay, so this is it. All right. So now we have our ideogram. You can change the colors. Okay, so if you look at the karyotype, let me just open it. I'll open it again for you to see. You can always change the colors as well. All right. Let's look at this. Let's just go back here and then look at it. So there's a karyotype. So you can see the chromosome names and labels. So if let's say you want to change one to a different color, you can try it. You can try it. Let's say you want to change this to a different color. You can try it and you can rerun. All right. But for now, let's look at the image. There's something I want you to see. If you take a look at the labels, all right, the labels here, the font sizes to me is small. All right. So it's possible to change the font size. All right. Don't forget that these things here is the ideogram, right? So the ideogram plotting and displaying every information is found in the ideogram.conf. So if you want to change something from here, then all that you have to do is to come to the ideogram.conf, which is it, and you can change it here. All right, so there's the label size. It's 40, but you can change it, let's say, to 80. This time using a text editor. All right, you can change it to 80. Then you can save it. All right, but if you don't want to do it here too, you can just come to your terminal. Let me just do it here. You can come to your terminal and say nano-ideogram.conf and then come to the label size here, then change it. So it was 40, but now I'm just changing it to 80. All right, so that is it. So once you are done, all that you have to do, let me just clear the screen again. All that I have to do is say circos-conf, and then you specify your configuration file again, and then you execute it. All right, so once you do that, then the old image will be replaced with the new one, which is the same name. And now you can see that the font size is now big. Okay, it's now large. All right, so this is how it's done. Okay, so now we have our ideogram, which has been displayed nicely for us. And so all that I have to do is to add the SNP distribution. All right, so that's what we are going to do next. So with the SNP distribution, the information can be found in the link file here. So this is it. All right, so I just looked at the SNPs and then just created a start end for it and then use it. Now the approach I'm using is the links. Links are usually used to visualize or display relationships, all right, but you can also use it in a different way, and that's what I've just done. For me, I'm not looking at relationship, I'm looking at SNP distributions, but then I just chose to use the link approach whereby I display it as links. Okay, but links normally are used to display relationships, but anyway, you can be intuitive and then use it in a different way, and that's what I've done. And so it's always up to you to decide what you want to do. But of course, still for beginners, it will take time to understand everything. But if you have any challenge, you can always reach out to me and then we can work out something. All right, so let's proceed. So I'll go back. I have my link file here. So I'll go back and then I am going to edit this again and then put in the additional information so that we can display it and have it like this. Okay, now we have this, and I want us to have something like this. Let me just go back. I want us to have this one like this, and then we can finalize it. Okay, so let's start. So to do that, we need to edit this, but I would like to make a copy of it and then use it. All right, so what I'll do is to make a copy. That's what I normally do. So I'll try to make a copy here. And this copy, I'll rename it as conf2. Of course, you can also do it on the command line. That's fine. Now I'm just doing the blend. Okay, a mixture of command line and GUI so that beginners get a hang of it. So what I'm going to do is to do this. I'm going to edit this one. I've made a copy because I'll still need the information here, and then I'll add on. All right. So I'm making a copy like this, and I'm going to edit it and then now include this link file, this information here. I'm going to include it here. All right. So that is why I'm going to edit this one. So this becomes conf2. Remember, I started with conf1, and now I'm going to Circos conf2 because I want to keep track. Normally, it's advisable to always make a copy, or let's say save the changes into different files, especially if it's going to be a major change, so that if something goes wrong, you can always go back to the old file. All right, so I'm going to edit the circos underscore conf underscore 2.conf. This time I'm going to use my text editor, and then I'll proceed. So this is what we wrote. Let me just remove some of the space. So I'm going to add something else to it. Okay, so here I'll go to this other line and then starts. All right, so I'm going to use links. Let's go back to Circos. There's a tutorial also for links that you can also check. So this tutorial is what helped me. So links is used to show relationships. I made mention of it, but I'm using this in a different approach, in a different way, which is also fine because that's how I want to display the information, the SNP distribution across the chromosomes. All right, so this is the basic syntax, the syntax for links, and then you can put whatever you want here. So that's what I'm going to do. I'm going to do something similar. So when I'm here, what I'll do is to first tell Circos this links information. So I'll say links, and then I'll end it. I'll say slash links, and then in that block I'll specify. Now with the links, you can specify one or more links. So here I just want a single link information because I have just a single file. So I'll put this also here. I'll say slash link, not this one, slash link, and then I'll end its link like this. All right, don't get confused. This is the main links block. Let me just put this space. So this is the main links blocks. So this is the mother. Okay, and then now we have, let's say, the kits. So there's a link. We can also have another link. That means we need this block again here. So in it, you just put in the information. So I'll start with the file equals, and I'll say link file.txt, and then I'll specify radius equals, I'll say 0.95R, and then color I'll say equals black, and that's for A4. Now the black, you can just say, you can use any color basically, but this black A4 I'm using it because I saw it in one of the Circos tutorials, so I thought I also use something similar. Normally I'll go for something that works first, and then I can later make modifications to learn more. So that's what you need to do. Always, if you are following the tutorial, don't try to change it to suit your needs. Try to follow the tutorial in its entirety. Don't change anything. Follow it line by line, command by command, and then after it is working for you, after it has worked, you can now gradually make changes to suit your needs. This is how you learn quickly and faster. All right, so when you are learning, don't try to immediately change everything. Learn everything as it is. Practice code by code, line by line, and then later make the changes. All right, now we also need to set the curves option. So I'll have it here. So curves modification. So here we will say Bezier radius equals 0.1R. So according to the Circos documentation, this value here, the Bezier radius, it works best, or it looks best if the value is small. All right, so this is what I'm using. Of course, this also is in one of the Circos tutorials, so I just thought I will adapt it here. And then thickness equals 1. All right, so that is what I have here. So basically, this is for the links. And of course, with the links, you can also set a limit. But now this basic command should work fine for us. So I will just keep them as it is. All right, so this becomes the basic information that we are going to use. All right. So now I'm going to save it. And then I'll display it by running Circos again. So this is it. This is the new configuration file. So I'll rerun Circos again here. I'll clear the screen and then rerun. So I'll say circos.conf, and this time the file is going to be conf2, not 1. So it's conf2.conf. So let's execute this command. Okay, so now Circos is doing its work, so we have to wait for everything to get done for us. So let's proceed. So it should be done very soon. It's done. So we can go back to it and then look at it. So this is it now. So you can open it and then you have your image here. So it's so cool. So this becomes the final image for it. Okay, so let's look at this image here. Now that you have this image, it's important to also interpret it. Of course, interpretation will also depend on you. It also depends on what information you want displayed and then how it has been displayed and all that stuff. So interpretation will be subjective. It has to be tailored towards what you want at the end of the day, and then that is it. So let's take a look at the image. Now, if you take a look at the image, let me just do this a bit. If you take a look at the image, the plot has been drawn in such a way that you can identify chromosome lengths as well. That also is important in some contexts. That has been done here on the ideogram. And then let's look at the black lines. Okay, so the black lines here in this case represents the SNPs. All right, so each of these lines here represents a SNPs. All right, so what you see at the end of the day is the SNP distribution across the chromosomes. All right, now with this figure here or plotted, you can see that some places are much darker than others. Some regions are much darker than others. For example, we have this, and we also have this one here. Okay, so darker regions will indicate the regions with high number of SNPs, okay, compared to other regions. All right. So for example, let's look at chromosome 7, right? We have chromosome 7. Now here we see that chromosome 7 and 6, these are different colors. All right, so we can change it. I think we even need to change it, and then we can just proceed. So there's a classical example of how we can change the color. So let's even change the color here, then we can talk more, because now these are merged together. So for beginners, it might be challenging, quite challenging to separate or distinguish between them. So I will change this. Let's just go back here. So I will change the color by coming to the karyotype. Let's go to 6. Okay, so here we can make it red. Then we can rerun Circos again. And then I will explain it. So I'll just rerun Circos so that we now have a distinct chromosome data. Okay, chromosomes distinct from each other with respect to colors, so that we can now explain it much better. All right, that has been done. So let's open the figure again, and then we can proceed. Okay, perfect. Now we have it done here. Okay, now we can proceed. Yeah. So I'll go over again. So we have the ideogram, which I made mention of which you can also see that by looking at them, you can see the length of those chromosomes. And then the dark, or let's say the black lines, all right, the black lines represent the SNPs. So what you see here represents the SNP distribution across the chromosomes. We have all of them here. So regions that are much darker indicates that those regions have high number of SNPs. So for example, we have this, we also have this, we also have this. Okay, now let's look at one of the darker regions, which is this one here. All right, so if you take a look at this one, for example, this is chromosome 7. We see that we have some dark areas here. Okay, so this indicates that with chromosome 7, this particular region, okay, there's a high number of SNPs in there. So if you compare this chromosome 7 to, let's say, chromosome 9, which is here, you will see that comparatively, chromosome 7 has a higher number of SNPs than chromosome 9, because here, even though there are dark regions, comparatively, this one is much darker. All right, so that's what you see here. Okay, so to further buttress this point, I want us to read the input file on the terminal and then look at the counts for the number of SNPs for chromosome 7 and chromosome 9, and then we can come back and explain it further. Okay, so we are going to read the input file, which is this one here. Okay, so this is it. All right, so here we can count. So OS1, let's go to the karyotype. So chromosome 7 is OS7, and then chromosome 9 is OS9. Okay, so we can easily count the number of SNPs by going to the terminal. Let's do an ls. We are going to count the SNPs for chromosome 7 and 9. We are using a link file, so I can just say grep OS7 link file. So I'll get all the information for chromosome 7. Of course, I don't want the detail, I only want the number. So I'll just issue this command and I'll say wc-l. So this will give me the number of SNPs for chromosome 7. I can do same for chromosome 9. So that's OS9. And I'll say link file and I'll say wc-l. So this gives me 296. All right. So now you can see we have 758,296 for chromosome 7 and 9 respectively. All right. So this tells us that the number of SNPs identified on chromosome 7 is higher. Okay, I think it's more than twice the number on chromosome 9. Okay, so that's what we see happening in this image that we see here. All right, so though using the numbers itself can tell you that chromosome 7, the number of SNPs on chromosome 7 is higher than chromosome 9. That's all that you can get. But then if you want the final details, if you want more details, then this is where the visualization comes in here. All right, so here we see that we can easily compare that. Okay, but using just the numbers alone, sometimes it's much better if you can visualize. So when you visualize, you can even know the exact areas where the SNPs have been concentrated. That is what we see here. Okay, so using visualizations like Circos can help you to really understand your data. It can help you to interpret. It can also help you to identify patterns easily. All right, it can help to identify patterns easily, and then that will help you to tell your story or present your story in the way that you want to present it. Okay, so visualizations can come in very, very handy, especially when dealing with dynamic data, and that is what we have just done here. All right. Now, it's also important to know that you can also generate this same plot using Galaxy. All right, so this is something that I'm going to work on. I think I've already worked on, by the way. So if you also want to learn how to generate Circos plots, or using Galaxy, then you can also check the video right here. All right. And also I have other tutorials on Circos. So if you also want to learn how to generate other plots using Circos, then you can also check this playlist right here. So that'll be all for this tutorial, and then I'll see you in the next session.